Okay, hello everyone. Welcome to the second of 3 in this advanced bioinformatics webinar series. Today we have our speaker who is Grant Roy. He is a research scientist in biomathematics at Singleron. And today he will be talking about MapResponse, Introduction to Machine Learning of Single-Cell Data. In his talk, he will cover the basics of machine learning classification models, connections to machine learning models to biological data, and also show a case study of a successful biomarker prognosis associations. So the talk will last around 45 minutes, plus we have 15 minutes of Q&A session at the end. So if you have a question, either raise your hand at the end of the talk, or you can either write your question in the chat and I will ask it directly to Grant. So without further ado, over to you, Grant.
Hi, thanks everyone for being here. So today I'm going to talk a little bit about machine learning in the context of single-cell data. So just to briefly overview, we're going to talk about just some general concepts of machine learning. And then I'm going to introduce a particular machine learning algorithm. And then we'll look at how we assess the importance of features and predictors. And then we'll look at a case study data of some AML data. And then we'll also discuss some extensions to some of these machine learning methods that can be particularly useful in the context of biological data. So the setting of our problem is supervised machine learning. And what that means is we have a set of predictors, which in this case are going to be our genes. This is going to be our data matrix X. And what we're interested in is predicting Y, some response variable. In this case, it might be the state of a patient being disease or not disease, or whether or not that patient may or may not respond to treatment, right? So the data matrix X has a particular structure. If you work with biological data sets, then this is very familiar to you. And it's also familiar probably that we can do all sorts of pre-processing on the data. And this includes things like normalization. This can even include, and usually does include, things like dimensionality reduction. However, we're going to use machine learning actually to do dimensionality reduction. So we're not going to reduce the dimensionality of our data set as we feed it to the machine learning algorithm. And so what we want to do is split our data into different sets, right? So we're going to have a set that our algorithm will learn with. We'll test it on a portion of that data set, but we'll also have a completely separate data set that at no time enters the training process that we want to validate the algorithm on later. Right? And ideally, we'd like that validation portion of our data to be as representative of data that the algorithm might see in the future, right? Or as difficult as possible, as we can make it with the data we have on hand. Okay, so with single cell data, these data sets come with a particular structure. And one of the things that we want to pay attention to, which you're probably familiar with, is the high dimensionality of this data set, right? So if we say P is the total number of predictors, or the total number of genes in our case, then we know that P is very large, right? And particularly in the case of bulk data, N, or the number of samples or the number of cells, can be much lower in many cases than P. So formally, we have an underdetermined matrix, meaning we have more predictors than we do samples, right? And so whenever this is the case, this can be an issue for predicting on that data. Further, another issue we have is that most of these entries are actually going to be 0 in the case of single cell data. So that gives the matrix a sparse structure, right? And in the case of single cell data, we actually do have a lot of cells in a lot of cases, but we typically don't have as many as we would like, given how large P typically is, right? So there's still a lot of challenges robustly learning from this data in a way that's principled, I think. So we'll look at a couple of particular classes of models here. And one of these canonical models is based on a tree model, right? And that is essentially looking at a single value of a feature and then looking at another succession of those values, right? And so features that appear along the same traversal path of a tree are actually features that are interacting, right? So this model allows you to capture some feature interactions. Okay, but the problem is a single tree is actually has a high variance, and it can be very susceptible to changes, small changes in the data set, et cetera. So what we can look at a single tree as is a weak learner. What that means essentially is it's only slightly better than the baseline of predicting some response variable. So if we had a set of classes where the response variable was evenly distributed, like 50% was one class, 50% was another, a weak learner would maybe be like 53, 54, 55% accurate. It would be slightly better than you could do just by guessing. What was noticed early on in the computer science literature is that actually you could combine these weak learners into something much more powerful, right? And that by actually combining these weak learners in a principled way, you get something very, very powerful, right? And that's what, you know, random forests and boosting are essentially, right? They're ways of combining these weak learners into ensembles that are far more powerful than the individual components. All right, so this is a very powerful idea from machine learning, and it's had obviously a lot of impact throughout. So what we want to do, though, is kind of understand what the complexity is to some degree of the method we're going to use on the data, right? Because this is going to help us understand more about what our algorithm is doing, right? Especially when we start to interrogate what kinds of predictors are important for a particular model, right? So on the left here, we have a decision stump, which is simply a tree of only depth 1. And what this means is you can't really model any interactions. There's simply one split, and that's it, right? When we start increasing the depth of this tree, we can capture more and more complex interactions in the data set. We have more paths that can be traversed, right? So as we increase the complexity of this model, we're increasing its capacity to fit a data set, right? Now, this can be both good and bad depending on how we do that. Okay, so all the way to the right here, we have, you know, what would be considered a deep tree, right? If we have a tree of maybe depth 15 or 20 or something like that. Okay. So what's interesting now is that we have the ability to compute some values for the predictors that we find in our model. So one of the downsides of using boosted models for a long time is that they were considered black box learners, and it was difficult to kind of interpret what they were doing in terms of which predictors were informative on a data set. So as we increase the complexity of the tree, we can compute these values of feature importances for different levels of complexity, right? And what's interesting is that decision stumps are the simplest case of the tree. And when we compute Shapley values for the decision stump, there's evidence here that these are actually very consistent, and we're getting an idea of what maybe a linear model might see, right? And then as we add complexity to the tree, you're looking at features that may be interacting. So what we see as we do this is very few predictors actually have significance at all in a decision stump. And then we see as we add complexity that these features start to interact a little bit more. And we can see that in the Shapley values that we're calculating here. So for the tree depth of 1, they start to zero out after about 15 or 20 values. Right? And then they'll have lower and lower values as we go down in the different depths of the trees. Okay, so essentially what we're trying to do is we're trying to study how increasing model complexity interacts with, one, both the accuracy of a prediction, but also arguably more importantly, or as importantly, what the impacts are on our computed Shapley values from these trees, right? And the Shapley values, just to kind of go over that again briefly, these are estimates of each variable's importance to a prediction. In other words, which features are driving the prediction, or most important, or having the most significant impact on making that prediction. These are the features that are most important. And so one way to look at that, which we just saw, is you can look at those values as a result of increasing the complexity of the tree. Right? So we don't just train a single model and compute some values for the variables. What we really want to do is train several different models at several different levels of complexity and then examine what the features look like relative to each level of complexity. That's one way we can look at this. Another way we can look at this on the right is actually starting with the full data set where P is all of our starting genes, right? We start with all of the genes as predictors. We train a model, we compute the Shapley values, and then we selectively remove the most important values at each iteration. So we recursively eliminate these features from the data set. And so what we get then, as we do that, basically what we're training is an ensemble of ensembles at that point. Because what we're doing is we're taking a single model and then we're removing the most important genes at each level. And we're doing that repeatedly for some number of iterations. Right? And so what's interesting is that if you do this, you actually increase the accuracy of the overall prediction if you combine these ensembles at the end of all of this. Right? So we might start with one here in blue that has all of the features. Then we have one with 25 features removed, one with 50 features removed, etc. And as we do that and combine them, then overall the model is actually better, right? The important point, though, is that when we do this, you want to predict the probability of a class being a certain, the probability of a class and not just the label, right? And the reason you want to do that is that predicting the probability of the class strictly gives you more information about the label than if you were just predicting the label itself. So when you combine these, each model will give you a probability estimate, and the more sure one model is, it's going to have more weight than another that's not quite as sure. Okay. Sorry. So what we sort of talked about right now is we've talked about XGBoost as a tree model, or boosting as a tree model. But we've also talked about training successive models in a way where we eliminate some features or increase complexity, starting from a baseline complexity and train more complex models. So most of the time when you hear XGBoost, you're automatically going to think that these are boosted tree models, right? Because that's almost always how it's used. However, XGBoost is just a library. It's not, there's a family of algorithms here that are actually available, right? And so one that we think is particularly useful for single cell data is L2 boosting, which is essentially a boosted linear model, right? And the question would be, why would you do that versus a tree model? Well, I think in the context of machine learning generally, what we're really assuming when we do this, when we train a machine learning model, what you're assuming is essentially that there's some low dimensional structure within your high dimensional data set that contains a sort of, the true model of what's actually happening. In other words, not all of your genes are relevant to a certain predictive task, right? There's some low dimensional subset or combination or interaction that's actually all you really need to find in your data set to be able to do prediction or to model that data set. And so these linear models, these boosted linear models, or L2 boosting, actually have a nice property where we can strictly regularize them in a way that lets us try to recover that low dimensional structure, right? And so I think that's very important in single cell analysis. Okay. So what does this mean? So basically, we're viewing this L2 boosting as an optimization problem, right? And we're going to minimize, the L1 norm subject to these constraints. What that means basically is we're going to take P, right, which is the number of genes we have in a data set. And what we want to find is actually the sparsest solution that's still predictive of Y. So this first row here, we might have, 3 genes, the first 2 and the third, that are kept, so to speak, in the model, right? And then the bottom, we have 1 gene that's kept in the model, right? So the bottom row is sparser than the top one. And this is actually what we want in a good model, right, of a data set. We want to find the sparsest solution that's still predictive of the data set. Right? There might be many solutions to a particular prediction problem, but what we're actually interested in is if we can find the sparsest one. Okay, to look at a case study here for some AML data, we have 4 batches of data, and these are processed at different times, right? So there could and probably are very well be batch effects. And then we want to predict, based on the single cell RNA profile, whether or not a patient will respond to treatment. Right. So we've trained models using the process described, where we're not actually training a model. I think that's probably one of the key takeaways. We don't train a model. We train several models, and they all have different characteristics because they all have different either complexity or they have different feature space. Okay. And so if we look at the accuracy overall, we're hitting about 90% accuracy on the cell level, and sample level accuracy about 80%. So there's one sample that's mischaracterized, but there are probably reasons why that appears that way. But overall, the model is very, very confident based on the scRNA profile of what these patients should be labeled in terms of responding or not responding to treatment. Okay. So another really important question, or I think critical question, we're looking at these data sets and building predictive models, is how can we measure the amount of information that we have in these predictors so that we're more sure of what they're predicting, and we have a better overall understanding of the data set. All right, so we can look at the same data, and we'll do the same thing where we're training and testing on different batches, right? So that's another key point, is when we do these predictions like here, we're not training and testing and splitting the batches equally. We're leaving out batches that probably have batch effects and not introducing those into the training set. So the model has to contend with a test set that's from a different batch that it hasn't seen during training, which could contain batch effects. Okay, so for this experiment, we're going to remove the top 25 SHAP features at each iteration, meaning we're going to successfully train several models, but we're going to remove features at each iteration, 25. And we're going to have a test set where the classes are evenly balanced. So 50% of one response, 50% of another response. And importantly, when we replace features, we're not going to just remove them. Or I'm sorry, when we remove the features, we're not going to actually remove them. We're going to replace them with randomized features, right? And so this is important because if we want to look at the information in the data set or how it's affecting a prediction, we don't actually want to reduce the dimensionality of the data set, because if our model is overfitting or it's using these extra dimensions to overfit, we don't want to remove them in this process. We actually want to just replace them with features that are purely uninformative, right? Okay. So another key point is that each iteration, what we're going to do is we're going to train the model twice and compute the Shapley values twice. All right, and then we're going to compare the Shapley values for each iteration. All right. And so what we see here is we have two different experiments where we did this. All right. The top one, we can see that over here on the left, the accuracy starts off pretty high, and then dips down. But it's always hovering a little over 7 or so, 70%. So feature overlaps by iteration. This means at each iteration, when you train the model and compare the two trained models at that iteration, the Shapley values, how much overlap is there in what it thinks are the most important predictors? And so as we start at the very beginning, here at the 0 iteration, at iteration 1, what we see is that there's a lot of agreement in the model about which features are important, meaning it's picking features that really do look significant, because for each time we train the model, the 2 times, the features are largely in agreement that it finds. But as we start removing features, the most important ones that it finds, then we start seeing the feature overlap drop way, way, way down. Right? And so we can see now at this point that the model is fitting noise. Right? And this, on this third row here, random features selected by iteration, what we see is that as we start removing features, the most important ones, the model starts picking out actually some of the random ones that we've put in. Right? So it's actually fitting noise in a sense. Right? So in other words, it's saying that the significant features I found are these, but they're actually the random features that you injected into the data set. Right? So we did this for 2 different runs. And the difference between the top row and the bottom row is that the bottom row actually has a proper level of regularization, which means we've given the model enough constraints that it's not over complex for the data set. And what we see is that the predictions of the model start approaching the 50% baseline, which is what we would expect if the model is no better than random guessing. Right? And that's what should happen as we remove features. Right? And so on the bottom right here, what we see is that the model up to 40 iterations, so that's 40 times 25 genes, it never selects any of the random features. Right? Up until a certain point here where you see this bifurcation in the graph in the middle on the bottom, what you're actually seeing here is an implementation detail of the boosting algorithm itself. Meaning after the Shapley values drop to 0, what actually happens is because of an implementation detail, it selects alphabetically the same features, which is interesting. But basically, when you get to about 40 iterations here, there's no more real information left in the data set. So the point of all this basically is to study how many features or what features in the data set in totality are actually relevant to the predictive task that you're doing, right? And also, how do you know that your model is properly constrained or properly regularized? We don't want to just look at predictive accuracy and then figure out, okay, well, this model is performing correctly based on it's predicting this or that. That's not really enough information to really have a kind of an understanding of what's going on in the model. But here we can tell by the regularization parameters we pick to train the model that the model on the bottom is much better, in a sense, more understandable than the one on the top. Okay. But what's maybe even more interesting is the question of, is overfitting a problem, and how much of a problem is it in a data set, right? So the traditional view in machine learning is that, once you start overfitting the data set, you start interpolating the training data, you're going to generalize poorly to data that you haven't seen. That's sort of the traditional view in machine learning. However, recently, this view has been called into question, and mainly so because of the large deep learning models that we have. And people started observing that even as you train these models to the point where they perfectly fit the training data set, they still tend to perform very well on data they haven't seen yet. And this is a big theoretical problem. And so there is some evidence that interpolating the training data set can be good in a sense. You can actually generalize better by doing that. And there's some literature in the boosting, but there's some boosting literature too that substantiates that as well. And the reasoning basically is that when you do overfit the training data set with some of these algorithms, or what happens actually is the noise in the data set actually tends to get isolated in some of these like further away trees. But when you combine them all together, in an ensemble, it actually smooths out the noisy points, and what you get is the signal. Right. So this is still ongoing, investigation in the overall field of machine learning right now. So noise in a data set is one thing, right? And for single cell data, one particularly thorny problem would be things like batch effects, or just any perturbation outside of, it could be sample collection or whatever, that is going to affect different data sets or different batches in ways that are orthogonal to the signal. In other words, what you have in the setting of machine learning basically is you have a situation where you're training your machine learning algorithm on a certain data set, but in reality, when you're providing it sort of data that's coming in, it's maybe more likely than not that that data is actually coming from a different distribution, meaning that it has different properties than the data that it was trained on. You may have quote unquote AML data, but if it's coming from a different lab, it's coming from a different hospital, this data is actually going to have a different distributional characteristic than data that you train on. Right? And there's sort of lots of ways you can try to address this, but what you're trying to do is account for the possibility of shifting distributions, right? And so we can measure the data that's coming in and test whether or not it fits a training distribution that the algorithm has seen, right? And so we can do a few different things like perturb the predictors while keeping one constant and see how much shift we get in the coefficients values for that predictor. Or what we can do is something like a projection norm, right? Where we've trained this model on the first data set. But what we do is we take the unlabeled data that's coming in and we assign it labels. We predict the values of that data coming in, and then we retrain the algorithm with that now training data set and measure how different the coefficients of our model are based on the data that came in. So we can have some idea basically of how distributionally robust the algorithm is. Right. And what we've talked about is this setting of ensemble models, right? Where we talked about training models of different complexity and training models with different predictors. But we can also train models that have different perturbations, right? And those perturbations would be perturbations we've used to simulate things like batch effects, right? And now we would have ensembles of models that have different distributional characteristics that we've trained on, right? And we can do the same thing at that point and combine those. And overall, we have a more powerful model than we would otherwise, right? And again, there's some literature that suggests that we don't need to train or test the model on the same data set it was trained on, right? The same type of distribution. We can kind of engineer a distribution, if you will, or maybe several that are slightly different, and then combine these predictions to get an overall better prediction. Right? Okay, so that's the end of the presentation. Thank you.
Thank you, Graham. That was really interesting talk. So we have a few questions on the chat. So the first one is with regard to number of features and number of samples, does it matter if number of samples are much smaller than number of features?
No. So that's one of the advantages of this kind of L2 boosting approach, right? These are methods that are specifically designed for those kinds of underdetermined matrices, right? Where you have less samples than you do predictors, right? So that's actually not an issue for these types of algorithms.
Okay. Another question is, do you have a good guideline on how many genes to reduce every time? Will 20/20/5 be a good stepwise reduction on number of features?
Yeah, we don't have necessarily a guidance on that, but it's more of actually at this point a heuristic where we want to take away enough so that the iterations, aren't excessive. Right? We could actually the best way to do it would probably be to just remove one gene at a time. Right? And if the runtime of the algorithm isn't that critical, we would have more information probably if we just did it, one at a time for however long it took. But that would extend the training time quite a bit, which, depending on your setup, may or may not be like a big concern.
Okay. Another question is, what are the differences between top SHAP features and top important features?
So the Shapley values are a particular way to calculate important features. And these are basically based on ideas in game theory where you're trying to fairly attribute a share of a contribution, basically. And so it turns out that the predictors you can view them as playing a game, and then use this idea from game theory to attribute basically how much of a share they have in a prediction. And so the Shapley values are one way of assigning importance to the features. There are some other ways that you can try to do that, but they're the only way right now that has like a sort of a good mathematical story of why we should trust them in a sense.
Okay. Another question is, will the shifting distributions be automatically picked up by trees or L2 corrections?
So no, we have to purposely train ensembles to do that. Basically, we have to engineer an ensemble that's seen those kinds of shifts in distributions. So there is a bit of work involved in trying to do that, right? Which is you're basically trying to engineer a set of perturbations that capture what your model was likely to see in the field.
Okay. And one more question, is there any way to overcome if the training samples are very different from the test samples? Are different feature selections strategies recommended with similar or very different training and test data combinations?
So one thing is, you can only overcome so much difference. In other words, if you have a completely different distribution, then really you're talking about two different problems, and we can't really expect our model to be able to adapt to any distribution, right? But the idea is, right now, what a lot of people are doing in machine learning is just building a model, and like somebody gives them some data and they predict it, right? But what we really want to know is not just what the predictions are, but really how reliable they are. And so we can measure these distributions in a lot of different ways, and we can use a few different strategies to know what we should expect the model to be able to predict or not. And so we can give a reliability score. Okay, so the other part of that is we can adjust. So there are some ideas where we can think about things like self-adaptive models, right? Where let's say I've trained my model on like 20 different distributions. Well, that's actually several different models, but what we can do is, as the data set comes in, we can adapt to that data set, right? And so there's another layer on the model, essentially, that's potentially self-adaptive, right? And so you can deal with some shifts in that way.
Okay. We have another question. Do you use the raw count data or normalized batch corrected versions of them?
Yeah, so typically we'll do some normalization procedures on the data. But I mean, that being said, the models actually do perform fairly well, even just on raw count data, the UMI count data, even without normalization, but we do some normalization.
Okay, so that was the last question. If there are no more questions, then I would like to thank you again, Grant, for staying up late for giving this webinar. So this talk has been recorded and will be uploaded onto our YouTube channel. So thank you everyone for participating, and see you all soon. Bye.